Okay, good day, everyone. Like I promised, we will be discussing or we'll continue with the discussion of the synthesis of urea. In the last discussion, we talked about how this amino group from the various amino acids were collected in form of glutamine, and then glutamine undergo oxidative deamination to produce ammonia in different cells, different organs. We have seen how this ammonium from different cells and different organs were all collected in form of glutamine and in form of alanine, then taken to the liver. Then the ammonia is released. Then you have also all the glutamine in the liver taken into the mitochondria, where it is undergo oxidative deamination to release the amino group. Thereby, you have all the amino group concentrated form of ammonia in the mitochondria. So please read. I expect you to go and while reading, read on transportation of gluten from the cytosol to the mitochondria. Okay. So today we will talk about the synthesis of urea. Human beings, we said that the ammonium can be excreted in form of urea. How is it? How is? Of course, in the process, we will see, for the case of the ammonium, in the case of aquatic animals, we say they can release it in form of ammonium, which would not need explanation on how it is released. It's ammonia that is released. And for humans, most terrestrial animals, we say urea, which we'll see how the urea will be produced. And in the case of uric acid, I'm very sure in the degradation process, we'll see how maybe uric acid is produced. I want to do it, so we see how uric acid is produced. So definitely cover everything. So urea synthesis was first discovered by Hans Krebs and his student Hans Litz and his own supervisor discovered it in 1932. This Hans Krebs is the same person that discovered TCA cycle. I think you can remember it is also called Krebs cycle. So it's the same person. You see, it's just before the discovery of Krebs cycle, and we'll see how this cycle is even also related to Krebs cycle. That leads to even the naming of Krebs bicycle. Sometimes it's called the Krebs bicycle, which referring to Krebs cycle and the urea cycle, or the TCA cycle. Cycle. So the cycle is a cycle that takes place in the kidney. Sorry, in the liver. In the liver, between the cytosol and the mitochondria, the reactions take place there. All the reactions are 5. Although some textbook will tell you the reactions of the citric acid cycle, they are 4. But generally they are 5. So we'll see the first one, which is not included or counted. We'll see what happened. And we'll see in all the cases what happened. But basically, all the reactions, they are part of the urea cycle. The first reaction is considered not to be part of the cycle, but nevertheless, we'll see how the reaction takes place. The 2 of the reaction, including the one that is not counted, takes place in the mitochondria. And then 1 of the reaction, 3 of the reaction counted takes place in the cytosol. Why this one is not counted in the reaction is because it is not involved in the cycle directly. It's not involved in the cycle. I mean, we can see the cycle here. See, you can see my cursor. You can see where this post-reaction, the ammonium that is being produced is converted to carbonyl phosphate. The carbonyl phosphate now is involved The reaction with ornithine to form citrulline. Citrulline goes out of the mitochondria to cytosol to form citryl AMP. Citryl AMP condenses with aspartate to form adenosine. Adenosine is broken down to form fumarate and arginine. And arginine is also broken down by arginase to form ornithine and urea. Then the ornithine is transported back to the mitochondria through ornithine transporter, and then also condense with another carbonyl phosphate to form another citrulline. And citrulline will be taken out of the mitochondria through citrulline transporter. And then the citrulline in the cytosol will also combine with ATP to form citryl AMP. Like this, you will see each of these steps, the enzymes involved and how does the reaction takes place. Okay, so this is basically what I explained, and the reaction, when we look at the whole reaction, is urea synthesis. And what happens is in the urea synthesis, we have only two ammonium that react with carbon dioxide to give urea. And we can see from the structure of the urea, we have a carbonyl carbon and then the amino group, two amino groups. These two amino groups, we'll see where are the two amino groups coming from. What are the contributors of the amino groups? The contributors of the amino groups actually the ammonium that comes from all these amino acid deamination. And then finally, with the formation of urea and water. So we'll see that, and then two hydrogen protons in form of a reducing equivalent. We will see how we included the energetic of the process. Okay. Now, okay, no. Yes. This is the first reaction. Like we have already explained, now the first reaction means the ammonium that is produced in the mitochondria will now react with CO2. Now, somebody would ask, where is the CO2 coming from? The CO2 comes from, it is mitochondria. And in the mitochondria is where the citric acid cycle takes place. And we know the citric acid cycle releases CO2. So this could actually come from the citric acid cycle. So in form of carbon dioxide, this carbon dioxide in form of bicarbonate because it's in the solution. So it reacts with ammonium in the presence of ATP because the two molecules cannot just react. The energy is not enough for them to form. So one ATP is used to bring the ammonium and the carbon dioxide to form what we call a carbonyl group. The carbonyl group is not also a high energy molecule. It doesn't have energy to also undergo the next reaction. Therefore, another ATP is also used. But so the first ATP now, it gives the energy. It releases the break of the phosphor and hydride bond in the first ATP releases energy, because I think you will do bioenergetics. You will talk about high energy molecules. ATP is included, and why even ATP is used the major source of energy in metabolic reactions. So the first ATP now undergo the breakage in the phosphor and hydride bond. So the breakage in this bond releases high amount of energy. We know that bond breakage releases energy, bond formation uses energy. Only that some bonds have higher energy than others. So in this phosphor and hydride bond is high energy. So once it is broken, it releases the energy. So that energy is being captured, it's housed in the formation of the carbonyl group, which bring to the ammonium and the carbon forming a bond from the carbonyl group. Because the carbonyl group is not high energy and need to undergo the next reaction, which is the condensation reaction, which also requires energy. So another ATP is used, but in this case, when the phosphor and hydride bond is broken, a phosphate bond is formed, is attached to, or a phosphate is attached to carbonyl to form carbonyl phosphate. This bond is also high energy. It's breaking of this energy also, but this bond leads to release of energy, which can be used for the next reaction. So basically, this first reaction consumes 2 ATP to produce 2 ADP and 1 inorganic phosphate. But what happened to the other inorganic phosphate? It is attached to carbamyl group to form carbamyl phosphate. And this reaction is catalyzed by carbamyl phosphate synthesis. We call it carbamyl phosphate synthesis 1. You will see another isoform of this enzyme when you are discussing nucleotide metabolism, synthesis of remnant. You came across carbamyl phosphate synthesis 2, which is catalyzed reaction similar to this. Okay, so what happened? So this is the first reaction. In the first reaction, remember the other one we said is not counted as the first reaction. This is the first reaction now. And from here, you can also see this part is the first reaction, the one we just explained. And this is the carbamyl phosphate we mentioned. You have the amino group here from the ammonium and the carbonyl group from the carbon dioxide. And this is the phosphate that is added to form what we call carbamyl phosphates. The next reaction is the condensation or the transfer of the carbamyl group to ornithine. Remember, we said ornithine comes from the cytosol. It is transported through ornithine transporter into the mitochondria. And once it comes into the mitochondria, it's now reacting with the carbamyl group from the carbamyl phosphate by an enzyme called ornithine transcarbamylase. This leads to formation of what we call acetylene. And we can see how the reaction takes place between the carbonyl group of the carbamyl phosphate reacting with this amino group of ornithine. What is ornithine? Ornithine is an amino acid, but it's not a common amino acid. Remember the 20 amino acids we say they are not the only amino acid, but they are the common amino acids found in protein. But there are other amino acids that are not found in protein. One of them is ornithine. Citrulline is also one of them. So ornithine is an amino acid. So condensation of carbamyl phosphates or transfer of carbamyl group to ornithine, where you have the carbamyl group of ornithine reacting with the amino or secondary amino group of ornithine by the help of ornithine transcarbamylase, lead to formation of what we call citrulline. Remember, the reaction involves the breakage of this phosphate group, then release the form of inorganic phosphate. So we have citrulline being formed inside the mitochondria. So citrulline will be transported out of the mitochondria through citrulline transporter. And then let's follow the citrulline, see what happens. So the next reaction is condensation of citrulline with aspartate. Now the question somebody will ask, where is the aspartate coming from? Yes, aspartate is produced in the mitochondria. Remember, citric acid cycle takes place in the mitochondria. And when citric acid takes place, one of the okay, I'm just thinking whether to explain it here or because there is a point where we will definitely explain that. So let me just say one or two things. When we get there, you will see how they think on it. So oxaloacetate is a metabolite of citric acid cycle, and it also takes place in mitochondria. So when you have this cycle taking place and there is a lot of glutamate, there is possibility the oxaloacetate can also undergo transamination reaction using the glutamate, thereby the amino group in the glutamate can be transferred to oxaloacetate to form what we call aspartate. Aspartate can be we can undergo deamination to form oxaloacetate, or oxaloacetate can receive amino group 2 transamination reaction to form aspartate. So oxaloacetate is converted to aspartate by glutamate in the mitochondria. And this aspartate is being transported out of the mitochondria to cytosol and serve as the second contributor, second donor of the amino group in the synthesis of urea. Remember, when we see the structure of urea, we saw the 2 amino group and the carboxyl group. We've already explained where the first amino group comes from. And this aspartate is the second source of the amino group. And basically, this ammonium in the aspartate comes from the ammonium that is being released in the mitochondria. It is oxaloacetate that take this ammonium from the glutamate, and then the aspartate being transported out to the cytosol, and then in the cytosol, the aspartate will condense with citrulline. When aspartate condenses with citrulline, they form what we call adenosine. This reaction is catalyzed by adenosine synthetase. And I think you were told that once an enzyme called a synthetase, it simply involves the use of energy. Energy in form of ATP is involved. So how does this reaction take place? First of all, we have the reaction between, or activation of citrulline first. Citrulline is activated. I don't know, when you go back to the first cycle, they explain while introducing the urea cycle, you will see where exactly this intermediate is formed. First of all, the ATP is converted to AMP and the release of pyrophosphate, 2-phosphate. So the AMP is not just released, rather it is attached to citrulline. When you look at the structure of citrulline here, the carbonyl carbon of citrulline here, you can see my cursor. So this AMP is attached here to form what we call citrullyl AMP, which is an intermediate. That is why in this reaction it was not shown. But in the first reaction, I show in the summary of the pathway, you can see it here formed, and it's in bracket, the square bracket, which indicates that it is actually an intermediate, not a stable compound. Once it is formed, it also proceed to the next. So that is what exactly happened. It's AMP was cleaved, removed, and then this amino group of aspartate now attached to the carbonyl carbon. So instead of the carbonyl carbon now, the carbon is double bonded to oxygen initially before condensation with aspartate. Now the carbon is double bonded with the primary amino group of aspartate to form what we call adenosine. Now the next reaction is the adenosine will undergo another reaction, a cleavage. That compound will be cleaved, will be broken down to release what we call fumarate, and then arginine. Arginine is an amino acid. We all know arginine. So let's go back here. What happened here is, this is what actually happened here. This bond, this bond here between the amino nitrogen of the aspartate that comes in will be broken, and the alpha carbon of the aspartate will be broken down. Once it breaks, this will be released in form of fumarate, this group. While the other group, when you look at it, if you know the structure of arginine, this is the classical origin. So fumarate is formed, is released, and then we have arginine. This reaction is catalyzed by adenosine layers. And then the next reaction, we have arginine Now, hydrolysis of arginine by an enzyme called arginase. Just in the presence of water, arginine is broken down to release urea, and then ornithine. When this is broken, this bond is broken, you can see this is now our ornithine. So ornithine can go back to the mitochondria and also react with another carbonyl phosphate, and the cycle continues like this. So you can see the cycle happen without that formation of carbonyl phosphate. The cycle does not include formation of carbonyl phosphate. That is why some of the textbook, when they are talking about the reactions in the cycle, they only mention 4 reactions. The reaction catalyzed by ornithine transcarbamylase, then the reaction catalyzed by adenosine synthetase, then the reaction catalyzed by adenosine lyase, and then the reaction catalyzed by the last one is arginase. While the one catalyzed by carbonyl phosphate synthetase is not considered as part of the reaction in the cycle. So basically, this is just a summary of this the reaction. And we can see by numbering here, we have 5 reactions, including the first one, which we said is part of the cycle, but is still counted. Now, the next thing we'll see is the regulation of urea cycle. How is urea cycle? Urea cycle is basically, because most of the reactions in urea cycle is actually non-reversible reaction. So only when you have a problem in any of them, you can have an accumulation of the substrate. But the first enzyme is involved in regulating. And this enzyme is being induced by what we call another molecule. Which enzyme is that? Carbonyl phosphate synthetase 1. It's being induced by N-acetyl glutamate. Once there is N-acetyl glutamate, it activates this enzyme, the later formation of carbonyl phosphate, and then the downstream reaction will continue. But how is this N-acetyl glutamate also formed? The N-acetyl glutamate is also controlled. The formation of it is also controlled. It is controlled because it is formed between acetyl-CoA and glutamate. And we know once the acetyl-CoA, it is inside mitochondria. And if the citric acid cycle is happening, and there is high glutamate, glutamate can react with acetyl-CoA to form N-acetyl glutamate by N-acetyl glutamate synthase. So that N-acetyl glutamate, because we have high glutamate in the mitochondria. This glutamate is too much. We need to produce urea so that it is released out. The amino group will be released out so that the level of glutamate or the nitrogen will reduce. Okay, so the glutamate will react with acetyl-CoA because acetyl-CoA is available in the mitochondria. Then it forms the N-acetyl glutamate. The N-acetyl glutamate serves as the effector or serves as the inducer of the carbonyl phosphate synthesis, and the urea cycle will begin. And also, in turn, the N-acetyl glutamate is also being activated by the presence of arginine. And the accumulation of arginine indicated also the high accumulation of the nitrogen containing compounds. So it's also activated N-acetyl glutamate, and that also activates. This is how the urea cycle is controlled. Now the next, okay, this is also the same explanation. How is the urea cycle related to the citric acid cycle? Remember in the point where the third reaction by adenosine lyase with a fumarate is released. And this fumarate we know is the citric acid cycle intermediate. In the cytosol, citrate can be converted to, fumarate can be converted by fumarase to form malate. And we know malate can be transported back into the mitochondria. And by the action of malate dehydrogenase, oxaloacetate is produced. And oxaloacetate, remember, it is the oxaloacetate that is being or that undergo transamination in the presence of glutamate to become aspartate. And then this aspartate is the one that is transported out to mitochondria, and then condensed with citrulline to form adenosine. This is how the two cycle connect to each other, which substrate of one or metabolite of one is in use in the other one. We will see how does that affect the energetics. With respect to the energetics, we have fumarate form, and the fumarate can be used to produce malate, and the malate inside the mitochondria is converted to oxaloacetate, and we know the process involves the formation of the reducing equivalent in form of NADH plus H. And NADH plus H in citric acid site, in electron transport chain. I think when we are doing carbohydrate metabolism, we know that each NADH plus H is equivalent to 2.5 ATP. So in the whole urea cycle, 2.5 ATP is produced. But how many ATP is being used? If you can remember, the first reaction in the urea cycle, the first reaction involving carbamide phosphate synthesis, 2 ATP are used. In the second reaction, where ATP is used, is where? Is the synthesis of adenocystinate synthesis. Adenocystinate synthesis leading to formation of what? Where citrulline react with aspartate to give adenocystinate. And ATP is used. But to remember, the ATP is ATP converted to AMP, not ADP. So usually when you have a reaction where ATP is converted to AMP, the phosphoanhydride bond that is been wasted there are two. So they are counted as 2 ATP being used instead of 1. If ATP is used to produce ADP, meaning one phosphoanhydride bond is broken, you have ADP. The other phosphoanhydride bond is still there in the form of AD, diphosphate. But this one is monophosphate, meaning 2 anhydride bonds being wasted. So the 2 anhydride bind is counted now as 2 ATP instead of 1. Therefore, 4 ATP is used in urea cycle. So what are we trying to say by this? We are trying to say that the whole urea cycle, we know, is the breakdown degradation process. It leads to only usage of 1.5 ATP instead of the 4 ATP, because it's at the end or at a point produce fumarate, which the fumarate is converted to malate transported to the mitochondria and then converted to, I think you can remember the transportation of malate back to mitochondria. So with the malate aspartate shuttle, or there is a shuttle, I think, in carbohydrate metabolism where we discuss this transportation. You also need to go there, please, about that transportation. How does it occur? So malate is now converted to transported into mitochondria. And in the mitochondria, it's converted to oxaloacetate in the citric acid cycle. That conversion leads to formation of NADH plus H, which is equivalent to 2.5 ATP. 2.5 ATP minus 4 ATP, you have minus 1.5 ATP. The 1.5 ATP is the net ATP we use in urea cycle. Now, the defect in urea cycle. Basically, the defect in urea cycle could be one. One, in the sense that mostly any defect can lead to accumulation of any substrate, because the reaction is they are non-reversible reaction. So any enzyme having any problem, then the immediate substrate will lead to accumulation of that immediate substrate. But if the first enzyme is the problem, then that first enzyme can lead to accumulation of ammonium. So you have high ammonium, and we have made mention of the effect of high for ammonium. in the system and how it affects the source. So when it involves the high ammonia, because of course when it involves any of the enzyme, it can some of those enzymes, some of those substrate of those enzymes can accumulate. But some of them can be excreted, while some is difficult. Only that they are not easily excreted. They can accumulate, and that causes a lot of problem. But in the case of ammonia, there are several ways in which this can be bypassed. So number one, let me say, if the issue now has to do with the first enzyme that is involved in the activation of carbonyl phosphate synthetase. We know what is involved in the activation of carbonyl phosphate synthetase is N-acetyl glutamate. But then a drug in form of carbonyl glutamate can be given. This drug, this carbonyl glutamate, it looks similar to N-acetyl glutamate. So it can go and activate carbonyl phosphate synthetase. So carbonyl phosphate synthetase. If the issue now has to do with N-acetyl glutamate synthase, it cannot produce N-acetyl glutamate, which is the allosteric activator of carbonyl phosphate synthetase. So carbonyl phosphate synthetase cannot be activated due to defect in the in the N-acetyl glutamate synthetase. It cannot produce N-acetyl glutamate, which is an inducer of N-acetyl, which is an inducer of carbonyl phosphate synthetase. So a drug in form of carbonyl glutamate can be taken, and that carbonyl glutamate can go and serve as N-acetyl glutamate. So it will activate carbonyl phosphate synthetase, and then the cycle continue. Then the accumulated ammonium can be actually converted to urea. Another one is the excess ammonium can be removed out of the body. How? Some drugs can be given, or some aromatic compounds. Number one here, we have benzoic acid. When you take a benzoic acid, in the system, benzoic acid can be converted to benzoyl-CoA by reacting with coenzyme A. In the presence of ATP, it forms benzoyl-CoA. The benzoyl-CoA can conjugate with the glycine to form benzoyl-glycine. This is called hippuric acid. We'll discuss this when we come to, this is the classical drug metabolism, how drug is being metabolized in the system. So when you come to drug metabolism, definitely you'll see some of this example as phase 2 reaction in metabolism, with drug metabolism or xenobiotic metabolism. Another also is phenylbutyrate. Phenylbutyrate can undergo beta oxidation to produce acetyl-CoA and phenyl acetate. The phenyl acetate can also further react with the coenzyme A to form phenyl acetyl-CoA. Phenyl acetyl-CoA can react with the glutamine, and then glutamine to form phenyl acetyl-glutamine. Phenyl acetyl-glutamine can be excreted. So now we have the ammonium being excreted in form of glutamine and in form of glycine. So as it is excreted also, more of this phenylbutyrate will react with, will be converted and to phenyl acetyl-CoA, which react with another glutamine, and then phenyl acetyl-glutamine excreted like this, and then the ammonium group that accumulated will now be excreted in form of glutamine or in form of benzoyl glycine or hippuric acid, in the case may be, depending on which of the drug is used or which of the chemical is to treat the condition. So you have this being cleared. This ammonium that accumulated in the system can be cleared using either of these compounds. So please, if you have any question, do not hesitate to ask. And when I come next week, we will discuss this briefly. I am very much sure by then you must have gone through these videos and you have reserved your questions. If you have any question, we'll just look at the question and then we continue with amino acid degradation. We'll see how these amino acids are broken down. So see you next week. Please take your time, read, listen to these things, make a note from this jot. Start your reading right from now. Remember, I already told you the 6th week will be this week of our tests. The first CA test, I will pitch for this since you are at home, maybe for those of you that are not yet here. So make sure you read this, you started preparing for the test. So once we finish, we're going to have the test. Then I believe it will be the post course you do 300 level. All right, see you.